Hi, I'm Peter Rose. I'm part of the Spoke team, and today I will give you a technical review how we build and query the SPOKE-GeneLab Knowledge Graph. SPOKE-GeneLab is part of an NSF program called the Proto-OKN program, and our specific grant has a title Connecting Biomedical Information on Earth and in Space via the Spoke Knowledge Graph. My collaborators are listed below. Sergio Baronzini is our PI. We have our collaborators at UCSF and NASA, and Charlotte Nelson at Mate Bioservices. Mate Bioservices is actually hosting the knowledge graphs, and they're developing applications on top of Spoke. So let's get into it. So here's the outline. So first I'll give you an overview what Spoke is and a general overview about the Proto-OKN project. Then we get into details how we actually built the SPOKE-GeneLab knowledge graph, which contains space omics data. So what are omics data? Omics data, basically, we look at what happens to somebody who is in space versus on the ground. What kind of changes going on in terms of, let's say, transcriptomics, epigenomics, and so forth. Basically changes in the human body in space. Then we talk about how we can run a natural language query on this knowledge graph using what's called MCP servers. I'm sure you heard about agents and so on, AI agents. So this is one way we create an AI agent that can run natural language queries. And then we do a couple of demos to demonstrate the use of this. Okay, so what is the Proto-OKN project? So it's a large-scale effort by the NSF. It stands for Prototype OKN. So what's an OKN? It's defined on the bottom here. So it's an open knowledge network that is publicly accessible, an interconnected set of data repositories and associated knowledge graphs that will enable data-driven AI-based solutions. So that's what we're about. We're creating a knowledge graph, and then on top we build AI-based solutions. So the Proto-OKN projects have four themes: biology and health, environment, justice, technology, and manufacturing. And we are in the theme of biology and health. And as part of that, we are providing two knowledge graphs. One is Spoke. Spoke is a precision medicine knowledge graph that knows about human biology and diseases and health. This was already developed before the Proto-OKN project started, but we added some new features to it as part of this project. And then this is a collaboration with NASA. NASA provides GeneLab, which contains space life science data. Those are experiments run in space, for example, on the International Space Station. And GeneLab is part of NASA's open science data repository. And there's a link down here if you want to know more about the Proto-OKN project. So I will talk more about Spoke and GeneLab today, in particular focusing how we built the GeneLab knowledge graph. So what we're interested in, basically, we want to compare the risk in space versus terrestrial risk for human health. So as a human on Earth, we are influenced by the environment, pollution, by our diet, we have diseases and so on. And then in space, you have other risk factors like radiation, microgravity, and so forth. But there are also some commonalities. So, for example, if you are a wheelchair-bound person, you may get muscle atrophy. And that also happens in space because you don't have gravity there. So we're also looking at analogs for human space flight that you can observe on Earth and find how can we mitigate those space risks. Okay, so what are actually space risks? So according to NASA, they put this into five categories. First of all, is distance. You're far away, there's limited healthcare support for you. You're confined in a limited space, so your fitness is, you can't do much in terms of fitness. Your sleep is probably disrupted, you're under stress. Then you're in a hostile, closed environment. So if an infection spreads, there's really no way out. You are either in your outer space, there's microgravity, or if you're on a planet like Mars, or if you're on the moon, you have fractional gravity, and that affects muscles, like you can get muscle atrophy, vision changes, bone loss, etc., cardiovascular and immune issues. And then long term, through increased radiation, you increase your cancer risk. And those are kind of the risk factors we try to mitigate. So we try to understand how do those risk factors affect human health and what can we do about it. Okay, to answer those questions, we created two knowledge graphs. The first one is called SPOKE-GeneLab, incorporates data from NASA, and the goal is here to make space biology accessible to AI applications. So that Spoke GeneLab Knowledge Graph currently has two types of data, gene expression data and DNA methylation data, also called epigenomics data. And that's available across multiple organisms and tissues. Those are mostly experiments done, on the International Space Station. One thing GeneLab enables is a comparison between spaceflight samples and control samples like ground control. So, for example, they have mice on the International Space Station and on the ground under the same condition, the same temperature, the same food, etc., etc. And then we can see what difference does it make being in space versus being on the ground. How does it affect gene expression and so forth. Now, a lot of those experiments are done, for example, on model organisms like mice or rats, but we really need to map that to human health. So part of Spoke GeneLab is a mapping of those rodent genes to human genes. So that's there, so we can see what that effect is on human health. And to really look at human health effect, we can create a federated analysis or a query with our spoke knowledge graph shown below so that we know what is the effect of those gene expression changes on health. So that's why we connect to spoke. So for this project, we have a knowledge graph called SPOKE-OKN, and the idea is to connect space biology to human health. Now, this Spoke knowledge graph is not the full Spoke knowledge graph that's available at UCSF or by Mate Bio, but it's a subset of nodes that we extracted from this full knowledge graph. And we added additional information as part of this grant. One is we integrated social determinants of health and we linked them to known disease prevalence, at least within in the US. We're also interested in effect of environment. So we also capture environmental data, mostly from the EPA as well, and also the location where those contaminants are. And therefore, we have a whole geospatial hierarchy in this knowledge graph, going all the way from the state to the county to the city and finally the zip code level. And then to better understand health effects, we also have a mapping between gene and diseases in this knowledge graph. So those two knowledge graphs together, we can query them together and get an idea, how space risk factors, affect human health and maybe eventually come up with some mitigations for that. Okay, so what's shown here on the left side is the things that GeneLab provides. It has information about hazards such as gravity, radiation, confinement, etc. Then they ran analysis, which means they ran assays on it, like transcription profiling, protein quantification, metabolite profiling. Basically, those are assays to figure out what is changing if you're in space versus on the ground. And then the assay measures various variables, for example, the fold change in gene expression and things like that. Then on the spoke side, we can then map the organisms in, let's say, be it mice or human or something else. We can map the anatomy, what kind of tissue or organ was involved in this study, what cell type. We can map this to biological entities, such as which genes are up or down regulated, to compounds, to biological process and molecular functions. And then, Spoke knows all about diseases and symptoms, so we can map this to whatever, muscle atrophy and bone loss and cardiovascular deconditioning, etc. So this basically is the linkage between GeneLab and Spoke. So that was a little introduction what this project is about, and now we're going more into the technical part of the project. We'll show you how we actually build the Spoke GeneLab knowledge graph. So NASA's open science data repository and contains GeneLab, which has, those life science space flight data in it, and they provide an API. Through that API, we select data sets that are of interest to us and then download those data sets. And then we developed a custom pipeline that now takes those data sets, prepares them, massages the data, and gets them ready to upload into a knowledge graph. And as a knowledge graph database system, we use a property graph called Neo4j. So those data are loaded into a Neo4j knowledge graph, and that can be queried to a query language called Cypher, which I'll show you in a minute or so. So these are the data that are currently supported in this knowledge graph, transcriptional profiling, data coming from RNA sequencing and DNA microarrays. Those are the properties we measure like log2 fold changes. And we only include data that are statistically significant. So we have some selection criteria, what kind of data we incorporate into this knowledge graph. We also include, DNA methylation profiling data as well. And here we measure methylation difference of DNA. And we are currently preparing metagenomics data through amplicon sequencing. And in the future, we also include single cell RNA sequencing data. So here's a simplified knowledge graph about how the data are organized. So, for example, NASA conducts a mission. That mission conducts a study, or usually a mission conducts multiple studies. A study then performs multiple assays. And the type of data we are collecting here is one is we measure differential expression of a model organism genes, and the other one is differential methylation here. Then we map which kind of anatomical structures or cell types were involved in this assay. So we have an ontology mapper that maps the cell types and anatomy onto the operon or the cell ontology. So we have a common framework that we can use to integrate, with other knowledge graphs, for example, SPOKE. And then for the model organism genes, we map them to the human orthologs, and we have an ortholog mapper application that does that. So we can uh, basically take the data from the model organism and apply that to human health. Okay, so this is now how we can combine GeneLab and Spoke. So we have cell types, we have anatomy, and we have genes, and they can now be mapped onto the Spoke precision medicine knowledge graph. So we know, okay, this gene is related to particular diseases, it's related to particular proteins or compounds or drugs, and so on. So this is how we now connect spaceflight information to human health information. And combining the two together, that gives us now the power to analyze the space risk and maybe come up with some mitigation approaches. So one thing that's special about our GeneLab knowledge graph is that it not only contains data, but it also contains metadata. So this in green, this node in green, contains metadata that describes the knowledge graph itself. So for example, we have an assay node, and an assay has properties like factor one, factor two. That might be factor one, maybe spaceflight. Factor two, maybe ground control. It defines the materials, the technology that was used to run the assay, and so on. So it describes the property of an assay. Then we have edges or relationships. And for example, the measure differential expression relationship also has properties like an adjusted p-value and a log2fc variable or measurement. So here we have a description what those are. So basically the metadata, first of all, they define the schema. They define the node and relationship properties. So it's basically a self-defining knowledge graph. And the reason why we do that, it makes it AI ready. If you just say log2fc, the knowledge, the LLM may not know what that means, but here we have a definition for that. So that makes it AI ready. Now the AI can understand, what log2fc means, for example. Okay, now to query, this Neo4j knowledge graph, you use a language called Cypher. As you can see, it's somewhat human readable. It has missions and studies and assays and so on. But still, this is difficult. Even for me, that probably would take me 15 minutes to create this query. And that's really not suitable for, the general scientists. We can't ask them, to create those queries. So what we really need are natural language queries. Now, since, the Proto-OKN project started about 2 years ago, there has been a lot of progress with large language models and chat applications. So we started now applying those to this project. So when you do a query in an LLM or chat application, your chat application could be Claude, it could be ChatGPT. And usually the way you use it, you put in a prompt, let's say into ChatGPT or Claude. It will then go to the LLM and then you get a response back. Sometimes it may go to a website, retrieve information from the web and so on. But what we want to do here, we want this chat application get information from this Neo4j knowledge graph. So how can it access that information? For that, we developed a server that connects this chat application with the with this database here, and this is done through what's called an MCP server. What is that? It's MCP stands for Model Context Protocol. It's an open standard developed by Anthropic, and that allows a client like Claude Desktop or ChatGPT to connect to a database, and it's a standard, so it runs basically everywhere. So we developed a server called mcp-genelab, where you put in a natural language query that will then be converted into a Cypher query. It queries GeneLab, then returns the data back to the chat applications. It may iterate through multiple iterations here. It may run multiple queries to find, and once it has all the information it needs to answer your question, it will then, return a response to the user. And um, so, we for this demo we're using Claude Desktop. It has a number of LLMs available. By default, we use Sonnet 4.5. It works extremely well. It even has a better version called Opus 4.5. This is more for complex work. It's also more expensive. But we also use other LLMs. So, for example, we use Visual Studio Code together with GitHub Copilot, and that allows us to look at other LLMs, such as the Google Gemini, for example. Gemini 3 is their latest model, and the OpenAI models, again, GPT 5.2, the latest model. The new models, Gemini 3 and GPT 5.2, also do pretty well, but Sonnet 4.5 is still the best in terms of generating those natural language queries. Now, we created this MCP GeneLab server, and it has a number of tools. So the most important tool is obviously to run a query, a Cypher query. So that tool runs a Cypher query on our knowledge graph. Now, to run the query, it needs to really understand the schema of that knowledge graph. So we have this method, get Neo4j schema. So it will retrieve the schema, and then the LLM knows, okay, those are all the nodes, and it knows assays are connected to genes and so forth. So we'll get all that information. And then, as I told you, our knowledge graph is self-explaining, so it has metadata about. It has metadata about nodes, all the node properties, like what is a log2fc, and it has relationship metadata too. So you can get all those metadata too, and they provide context to the LLM so it understands the information that's present in that knowledge graph. Now then, since we're interested in space omics, we have specialized tools for GeneLab to analyze differential expression, to select assays, to create various kind of plots. And then we have visualization and reporting tools. So first of all, we have a tool that can visualize the schema, and I'll show you what that looks like. It basically creates a schema of the knowledge graph, and it has a feature called create chat transcript. So if you go through a session through multiple prompts, it will summarize those in a nicely formatted report. Okay, now we're coming to the demos here. Before I do the demo, let me give a little introduction here. So we're using Claude Desktop, and we need to tell Claude Desktop to use our MCP GeneLab server. How do we do that? We add it. So there's an edit config button here, and you can add an MCP server. It's very easy to do. There are two simple steps involved. The instructions are on here. I won't go through that here today. But you see, now we have SPOKE-GeneLab here, and we have also SPOKE-OKN here, ready to go, and they're running for the demo. And this is the first demo we're gonna do. I just first walk you through what we're gonna do, and then I actually run the demo. So this is the chat box in Claude Desktop. This is where you type your prompt. But before we do that, I want to make sure we're choosing the right knowledge graph. So if we go click on the plus sign, go to connectors, you see I selected Spoke GeneLab here. You can select multiple at once too. So here you can select which knowledge graph you want to search. You can also use other kind of services. For example, we can query PubMed if you turn this on. We'll show you an example later of doing that. So here you select which knowledge graphs do we want to search. And then first, let's say I want to get a description of the Spoke GeneLab knowledge graph. If I just type in at Spoke GeneLab, if I just type in the name, it will basically create a description. What is this knowledge graph about? What can you do with it? And then you may want to understand what its schema is. You could say, okay, visualize a schema, and it will generate a diagram, a graphical diagram. And at the end, maybe you want to put all of that into a PDF file. You can say create transcript. It will create a transcript that you can then save as a markdown file or as a PDF file. Now, I pre-recorded that demo and the results are available at this URL here. So if you want to look at it, you can go there and see that. But what we're going to do soon, we're going to run this live. But before we do that, as I said, we develop various tools. So for example, if you say visualize schema, you can see what it does. It says, okay, I need to create a schema visualization, but first I need to understand the schema of it. So it runs this get schema tool to get the schema. Then it says, oh, I need to also understand the edge properties. So let's run this get relationship metadata tool. And then finally, it creates what's called a mermaid class diagram essentially creates a schema. So you see it uses four tools basically to create the schema, and the schema looks like this. So this is the LLM auto-generated schema for the SPOKE-GeneLab knowledge graph. We have missions, studies, assays, etc., etc. So an assay is a node, and the node has properties. Those are the node properties. So an assay measure differential expression of model organism genes. So this is called an edge or relationship. And a relationship can also have properties like we talked about that before, like the log2 FC value, for example. That's called an edge property. So you see the LLM has a complete understanding of this entire schema, what type of information is available there. So it can use that, to answer any queries. And that brings us now to demo one. So I'm now going to switch over to use Claude Desktop. So here's Claude Desktop. If you're not used to it, it basically works just the same way as ChatGPT. And we're going to run a query here. But before we do that, let's check which knowledge graph we are querying. If we go to connectors here, we'll see I already selected SPOKE-GeneLab. So here you can select which knowledge graph you want to query or any other services. For example, we can also query PubMed and other services here. So first of all, to get a description of the knowledge graph, I can just put the name here, Spoke at SPOKE-GeneLab, and run this query, and it will give us a brief description, what kind of data are in SPOKE-GeneLab and what it's about. So, this is about NASA's GeneLab spaceflight data. It contains, spaceflight information like gene expression data and so forth. It tells you what kind of queries you can run, what kind of visualizations you can do, and maybe some ways how you could explore it. But to really understand this knowledge graph, let's first create a visualization of it. So we say visualize schema, and we get a graphical overview what the schema looks like. So we really understand what type of information is in there. So as I showed you in the slides, it goes through multiple steps here to create the visualization. First, now it gets the schema of the database, so it knows what kind of nodes and relationships are in there. Now it gets some information about metadata here, and now it's starting to create the diagram, the schema diagram, and it's gonna take a few seconds to create that. So it's probably hard to see here, but this is, the schema that it creates. Maybe if you remember from before, we had missions, we had studies, essays, and so forth. So here you see the essays with their properties, edges with their properties, and so on. So that's exactly what I showed you before. So this is really helpful. If you don't know what type of data is in there, then, it's hard to query the knowledge graph. So here you can see, okay, what type of information is available about missions or essays, etc., etc. Um, Now that we ran, this, I mean, this is a very simple prompt we had here, but let's say we want to save this. We can say create transcript, and it will now take this entire session here, including the prompts and the results, and put that into a nicely formatted document, which you can then save as a Markdown file or as a PDF file. So on the right side here, you see that is now the transcript that it creates. So you see that was my prompt, for example, SPOKE-GeneLab, and then whatever. This is a response that it gives, and it does that for everything we did. And at the end, it says, okay, which model did it use? It used that, Sonnet 4.5 model. So it keeps track of that also, which version of MCP GeneLab was served here. So this is a way you can save your work in a nicely formatted document. So now let's go back to the slides and go to demo number 2. Now this is a more difficult query and it will take too much time to run live here. So I pre-ran it. So I will show you the transcript. So this is the question we're asking. Assess differences in gene expression between spaceflight and ground control samples in the NASA study OSD-244, contrasting short and long duration missions. So what they did here, they have mice on the International Space Station and on the ground. We want to compare what happens to them in terms of their gene expression. And they do that at two time points for a short duration mission and for a long duration mission. Then we see what's the difference between the short and long duration missions. So let's look at the transcript. So here's a transcript. So first of all, I want to understand more about what is actually in the essay and what are the relationships here. So I just said, okay, SPOKE-GeneLab, describe the assay node and its relationship. So it describes what the assay node is. It has an identifier, what type of measurement was done, what technology was used, then what experimental factors were used, for example, spaceflight versus ground control, etc., etc. What relationships are there, for example, what anatomy and what anatomical structures were investigated or which cell types were investigated. And then it has the actual measurements there, like differential expression values like 2FC values or differential methylation data. But we really want to know more about this particular study, OSD 244. I said, okay, get me more information about study OSD 244. So this mission, I mean, this study was done by SpaceX mission 13, and those dates here, the organism was mouse. Here's the study objective. Here's the experimental design. There were 40 female mice that were used in these experiments. This is how the assay was running, an RNA sequencing assay, some details. And there are actually 34 different assays available. Now we need to choose one or two of those assays, but how do we do that? So that's why I said, okay, select assays for this study because there are 34 of them. How do I choose them? And this helps me to basically get an overview about the assay. So it says, okay, there are six types of experimental conditions. See, there's space flight at 30 and 60 days, and we have ground control at 30 and 60 days. So for the short duration flight, we're interested in the comparison between space flight and ground control at 30 days. So we choose number 5 and 3 here. And for the long duration flight, we choose 6 and 4 from this list here. And then I can just say, okay, we want to compare both. And then I just type in the indices 5, 3 for the short duration flight and 4, 6, 4 for the long duration flight. And then it gives me the two assays. As I mentioned, about 34 assays, but we are focusing now on those two. The first one is a short duration flight, spaceflight versus ground control. And the second one is this long duration flight, spaceflight versus ground control. And this guides you essentially through that process. If you had to do it yourself, it's probably very difficult to find those assays. So the tools in the server guide you through the process, and it actually suggests next step, what you may want to do. Maybe you want to find differential expressed genes for each comparison and other things. And let's do the first one here, find differential expressed genes for each comparison. So now it compares spaceflight versus ground control. So here are some upregulated genes with their log2 fold changes, downregulated genes. If you don't know what it means, don't worry about it, but you see it can now retrieve the data from this knowledge graph. And then there are particular ways differential expression data are usually plotted. One of them is called a volcano plot. So I said, okay, let's create a volcano plot here for the short duration flight, similarly for the long duration flight, and comes up with some summary. And then, we ran a 30-day versus 60-day duration flights. So ask, okay, what do they actually have in common? Which common differential genes do we have? So it does an analysis of that. And it tells us what kind of genes are involved and what kind of biology are they involved. In, involved in immune response in inflammation, coagulation cascade, etc., etc. What are those common genes that up or down regulated are listed here. And then we can also visualize the overlap between in pink, that's for the short duration flight, in blue is the long duration flight, and you see, how many genes are in common in terms of upregulation and downregulation. And then it summarizes some insight it got from that. But then, I want to make sure the data we got, is there some evidence in the literature? And as I mentioned, we also have this PubMed. We can also search PubMed through a service here. So I say identify publication that mentions these genes in the context of spaceflight or microgravity. And it pulled up, and like 10 or so examples of relevant publications that you can now read up more specifically about those genes, and how they're related to space health. And then at the end, I say create transcript. So this is how I created this document here. So it's very easy. You do your analysis, and at the end, you say create transcript, and you get a file like this. So that ends my demonstration number 2. And so let's talk about what our plans are for the next year of this project. We want to integrate more biomedical data. First of all, data from NASA. They have additional metagenomics data as well as single-cell RNA sequencing data from spaceflight experiments. So we will add those. Now, the folks at UCSF, they're looking at the electronic health records and see what we can learn there, especially in terms of what bone health. If you're in space, you have bone loss because there's no gravity, pulling on your body. So they're looking also just at the electronic health records to see what we can learn about bone health and try to integrate that into this knowledge graph here. Oops, sorry. And then for the MCP server that I just showed you that does the natural language queries, we want to develop a benchmark and to really evaluate it. Right now this is more, single examples that I showed you here. And then we optimize the server so, to make sure the results are correct, and we use the benchmarking results for that. And then, as I mentioned, our project is part of the larger Proto-OKN effort, which contains like more than 10 different type of knowledge graphs. So we want to look into how can we query across multiple knowledge graphs as well. We call them federated queries. And then also going beyond Proto-OKN by looking at other third-party knowledge graphs. I mean, there are many other knowledge graphs out there that we could query too. And there are already hundreds and thousands of MCP servers out there that we can also use. An example I used here was this PubMed server where we can query PubMed, but there are many other servers too that can be combined as well. The idea is you have multiple agents and they can help you, analyze your question basically and come up with some results. So what I showed you today is basically how we created this Neo4j property graph, how we can use an MCP server like mcp-GeneLab to run natural language queries on that using a chat application like Cloud Desktop or VS Studio or other chat applications using this MCP protocol. Now we'll have a second video where we show you where we also query an RDF knowledge graph. So the Proto-OKN project converts all knowledge graphs into one single framework called RDF, and that's queryable through a Sparkle endpoint. So we also developed another server called MCP Proto-OKN that can query all the Proto-OKN knowledge graphs. So in the next video, we will show you how we can query all the other knowledge graphs as well using that same kind of mechanism. So, in summary, I showed how we built the GeneLab knowledge graph, how we can use, new AI tools to query them. So avoiding people to learn a query language like Cypher, instead they can just use a natural language prompt. And over the 2 years, there's a lot of progress with the large language models, and this just will continue. So, the current models are not perfect. They're usable as I shown you in the example, but they will make mistakes. So, you always need to double check your results, but they're really a large help to get the information that you need. You just need to double check it. And, over time, this, will get much better. And that's the end of my presentation. Thank you.